Great. Well, welcome everyone. I'm really excited to talk to you today about milkweed cultivation and its impacts on detritivore diversity. So one of the research questions that I think is most compelling is this question of how agriculture and biological conservation interact with each other and are often opposing goals. And a classic example of this is between the milkweed plant and the monarch butterfly. And these two species interact with each other because the monarch lays its eggs on milkweed, and then when those eggs hatch and become caterpillars, the caterpillars use the milkweed plant as a source of food. Unfortunately, the expansion and intensification of agriculture has seriously impacted the milkweed cover throughout the United States. And additionally, many farmers specifically target milkweed because it has a sap that runs through it that's toxic, and it makes cows sick, and so they don't want it in their pasture fields. The reduction of milkweed throughout the US has seriously impacted the monarch population, which now don't have a place to lay their eggs. And this has become a large contributor to the decline of the monarch population. However, there may be a new agricultural opportunity that may be able to reconcile the tension between monarch populations and agriculture. And this is the market for milkweed floss. So as you can see here, milkweed floss comes from the seed pods from milkweed, and its biological purpose is to protect and help disperse the milkweed seeds, though this jacket company, Quartz, in Quebec, is now using milkweed floss to stuff down jackets. So as a note, this is not the first time that milkweed has been used commercially. In World War II, there was a campaign to have kids pick wild-growing milkweed pods, and they were using the floss to stuff in flotation devices, like the one this little boy is wearing here. And that was pioneered by Dr. Boris Berkman, who is an inventor in Chicago. So what is new about milkweed is this intentionality of cultivating it. So these fields provide an unprecedented opportunity for the regrowth of monarch butterflies and other biological diversity, including detritivores. So detritivores are a group of organisms that help break down litter, like roots and plant shoots and dead trees and animal remains. I've shown here some of the taxonomic groups of detritivores, including earthworms, spiders, centipedes, beetles, and pillbugs. And I'm really interested in detritivores as an extension of my interest in soils. So detritivores, by breaking down detritus and passing it off to microbial organisms, are kind of like a middleman in decomposition. And in agricultural systems, their presence has a huge potential to maintain soil fertility and perhaps lessen the need for fertilizers. So in this sense, their presence and diversity can also be seen as indicators of healthy soil functioning. So in terms of how agriculture impacts detritivores, tilling actually can be very detrimental. So tilling can break up soil structure, and you can see an example of a healthy soil structure here, which has lots of pore space between the soil aggregates. And detritivores make a habitat of this healthy soil structure inside of those pores. So milkweed is very promising in terms of detritivore diversity because it is a perennial crop. It lives for around 4 to 5 years, and so it has much lower tilling needs than a lot of annual crops like corn. So my objective for this project was to examine the composition of detritivore communities under milkweed cultivation in relation to other common New England land uses. So I did my research at Borderview Farm, which is in northern Vermont in Albert. The dominant soil type is Benson Rocky Silt Loam over limestone. And here I researched in a variety of treatments, including milkweed and corn. I also sampled in a perennial forage grass, which is interesting because it serves as a comparison between the perennial milkweed. And then I also sampled in forest as a control. So I used 3 different sampling techniques, and I'm not going to go into them now, but these 3 techniques target specific groups of detritivores, like worms, below-ground arthropods, and above-ground arthropods. And back in the lab, I ID'd all the specimens to the taxonomic species level where possible and otherwise used morphospecies. I'm going to talk about the results from the worm sampling and the pitfall traps in my presentation today. So the results for the worms. There was a surprising absence of worms in all the treatments, and this was really surprising. It was great. I thought it was great. But it was really surprising because we had trialed this method earlier in the spring, and we're getting around 10 worms per plot. So we were really surprised to find 0 worms, for the most part, 0 worms per plot. And this is perhaps attributable to the dry summer, which dried up the soil and made the mustard solution that we were using unable to penetrate the soil. We did find some worms, and we actually found worms in each of the sampling techniques. And so here I've just simply pooled all the numbers from each sampling technique. And you can see a clear and perhaps unsurprising trend that you get the most worms in forest and the least in corn, though perhaps optimistic that milkweed is has the second highest number of worms. So here are some results from the pitfall traps. In total, I collected 217 species and around 8,000 individuals. I've shown here species richness and Shannon's diversity. So richness is the total number of species per plot, and then Shannon's diversity is a metric that takes into account both species richness as well as the evenness of the number of individuals that are in each species. So you can see that milkweed, for both species richness and diversity, has significantly higher values than the other treatments. So very optimistic results. So here is, I've shown the average number of individuals per plot for certain kinds of taxonomic groups. And so you can see here that for springtails and ants, there is the highest number of individuals in the corn plot. So with 18, on average 18 springtails and 8 ants per plot for the forage grass. And then for the forest, we have the highest average number of harvestman spiders. And then for corn, we have the highest number of beetles. And perhaps this is surprising, though I want to make a note that plot is quite different from the species richness. So when we look at species richness, we actually see that milkweed has significantly higher values than the other treatments. And feel free to ask about that more in the Q&A. So we have very promising evidence that milkweed can boost Detritivore diversity, and I'm really excited to sample again this summer and get more data. Always great. I'm also hoping to do some additional analyses, including soil carbon and nitrogen for each treatment. And then I'm also interested in this idea of economic feasibility and long-term viability of the milkweed for farmers. So with that, a big thank you to the Gund Institute and the National Science Foundation for funding. Thank you to my advisor, Jillian Galford, and the co-PI of this project, Heather Darby. Kimberly Wallen and Nate Sanders were invaluable with lab help. Hannah Turner, Kathy Solerano, who's here, thank you, and Carolyn Bittner and Will Davis were very lovely field assistants. And then Roger Rainville and everyone at the Board of Refarm were very gracious in sharing their land with me. And yeah, thank you so much. Questions? Yeah.
This is really cool. I'm curious about, because you said worms generally, and are earthworms in that area? Did you look at native versus non-native species? What's the breakdown? And like when you look at richness, do you take that into account?
Okay, so I found that because we got such low values of worms, I ultimately decided that I wasn't going to break them up into species. I think it was mostly the Lumbraceae family of earthworms. I did not look at different kinds of worms. I was just looking at earthworms, if that helps answer your question. So if I got a bigger number of worms, then I think it would have been worth it to look at species. But because we got like 30 worms, I decided not to. Other questions? Sorry, Kimberly. Okay, Hillary.
So do you have any sense of that third question about the feasibility? This is something we expect to see expanding in Vermont?
Yeah, so great question. In terms of economic feasibility, the farmers get more money per acre of milkweed than they do of corn. So they get around $800 to $1,000 per acre, whereas corn usually yields $600 per acre. However, there's a bit more variability in the yields, just because we don't have a standardized method of growing milkweed like we do for corn. So there's a little bit of risk there. And there's some other aspects of milkweed cultivation that also can make it more expensive in terms of labor costs. So whereas corn, you use mechanized equipment to harvest it, milkweed, what they did this year at least, is they manually went in the fields and cut off each seed pod. So that can be really expensive in terms of labor. Yeah. Kimberly?
Do we have time?
I think so.
Yeah, I was just wondering, are you planning to look at functional diversity?
Yeah, great question. I am still figuring that out, but so the idea of looking at functional diversity is really important to me because it can help connect diversity to actual ecosystem service provision. So that would be really interesting to see, okay, we have more diversity and perhaps functional diversity in milkweed. Does that actually translate to better nutrient cycling in the soil? So that's one of my main research questions, and I'm still kind of figuring out how I will do that, but it is a priority, so thank you. Yeah, Alan.
I guess sort of along those lines,
Mhm.
and maybe it would go hand in hand, is thinking about looking at biomass of the different groups.
Yeah.
Because there's going to be something aligned with metabolic rate in that case, and it might be helpful to
Yeah.
have those services.
That's a great point, and that's something I'm really interested in doing, because if we go back to this, it's pretty interesting because the corn, as I said, had a huge average number of beetles. And so beetles are pretty big when you're thinking about Detritivore. It's much bigger than springtails and even a lot of the ants. And so I think the biomass will definitely be weighted in terms of larger animals for the corn, whereas for forage grass, just by observation, I can see that there's a diversity in terms of biomass. So that, yeah, I would love to look into that. Thank you.
Okay, that's our 3 minutes for questions. We still have, we have 2 minutes of transition, but if you'd like to stay in this room and keep asking questions, please feel free.
Thanks.